Thank you so much for watching this tutorial where I'll go through the workflow necessary on KNIME to use it as a pharmacovigilance tool to monitor the adverse events of a special patient population. So to start off with, we downloaded our data from the Vaccine Adverse Events Reporting System, better known as VAERS. And so when downloading the data, it comes as three different tables. And so the first table contains information about the patient's demographics, looking into what the age, sex, as well as past medical history and any current medication the patient is taking. Our second file data contains information about the vaccine received. So looking into who the manufacturer was, what type of vaccine was it, where was it administered at, and our last set of data contains information about the symptoms and adverse events that were reported from receiving the vaccine. And so just to open up one of these nodes and data tables, I will open up the vaccine uh, table just to show you what exactly that looks like. And you'll see here how it contains information about the manufacturer and the type of vaccine. But I really want to draw your attention to this column called the VAERS identification. And so that is going to be a identifier without giving the patient's name, but each report that is generated comes with an identifier to align the three different tables together. And so instead of analyzing this information with three different tables, it only makes sense to join them together. So using the joiner nodes, we're able to do exactly that. And that was to identify and align the different tables and rows together by that VAERS identification number. And so moving forward in our workflow, we wanted to go ahead and clean the data. And so it was necessary to clean the data because the VAERS operates and runs off of user-generated data, meaning that I myself can go in there and report an adverse event, a nurse or physician can, and as well as someone who works with a drug manufacturer, they could go in and generate a report. And so when going through cleaning the data, we wanted to remove any columns that really were not necessary for our analysis, and also removing any missing data, just so it was nice and easier to work with. But moving forward in our workflows, the really powerful tools with KNIME came in the form of string manipulation tool. And taking a step back of our research question and proof of concept is that we really wanted to see what adverse events have been reported for patients who have multiple sclerosis, but also receive the COVID vaccine. So we were able to filter out all of the vaccines um, that had adverse reports generated that weren't pertaining to the COVID vaccine. So any of the flu adverse events or the shingles adverse events, those were filtered out. And we wanted to identify our special patient population. And so with multiple sclerosis and how the information is user generated, we really wanted to standardize the spelling of multiple sclerosis. So using the string manipulation node, I'll show you how exactly we configured it to do that. Using the reg replace function, we were able to go into the history column, which contains information about the patient's medical history, and standardize the spelling of multiple sclerosis, whether it was the formal way, uppercase or lowercase, and the shorthand way, uppercase and lowercase, and standardize it to MS, the uppercase shorthand format. And it created a new column um, given that spelling. So from there, we wanted to identify which rows contain that new spelling of multiple sclerosis. So we once again use the string manipulation node, and I'll show you how we configured it using the reg matcher function. So now we wanted to go through that new column that contains the standardized spelling of multiple sclerosis, and we wanted to see if there was MS standing in that column. So that's how we configured it. And so just to show you what the exput was for using that function is that you'll see in our new column here, it says false. So the false is telling us that when going through that column there, it was not able to match the spelling of MS. So given that, we asked KNIME to filter out all of the columns that contain false. So we would only see the columns where um, it would be true. So once we did that, we would have our special patient population of interest. So if I go back over to our new column here, instead of saying it false, you'll see how it says true. And then moving back over to our history column, which contains information about the patient's medical history, you'll see how exactly we do have multiple sclerosis in this column. And you can also see how I alluded to the different spellings in MS, whether it be shorthand or the formal way, uppercase or lowercase. You can see how exactly there is that variety of spelling of multiple sclerosis. And so now that we were able to identify our special patient population, um, the next part of analyzing the results. So that's where there is a great a deal of flexibility of how you go about analyzing results, really going back into what your research question is and how you go about getting there. With this being a proof of concept idea, really want to do baseline and do quantification methods. And I'll go through the quick steps of how we went about doing that. So in the second workflow, um, we went about going through analyzing the data. And so when analyzing the data, we wanted to use the Excel sheet, which the data was exported to, and bring it back, and then combine our symptoms. So the way that VAERS is generated, the symptoms are broken down into 3 different columns where the symptoms and adverse events were reported, but it made sense to go ahead and combine them all together into one column. And then we wanted to configure the data in a format so that it would be compatible with the bag of words creator node. So to do that, we had to use the strings to document node as well as the document data extractor node. And so the bag of words creator node I found extremely useful for the quantifying methods because it broke down those symptom reports into a much easier digestible way. And see if I can open up the outputs that was generated from that node. And so you'll see here how in this term column, how it's just single phrase words. And it makes it easier to do quantification methods compared to when you see our document body text, how we have these long sentences, and it's not quite as easy to do quantifications of these terms. And so when looking at the exput from our bag of words creator node, you'll see how it's able to identify words such as anaphylactic, um, whether it be fall, pain, feeling. These are things that we can quantify and build out. And so I was able to export this data back to Excel and create a bar graph quantifying how many times certain terms showed up, and as well as creating a pivot table, as well as another way to analyze and view the data. But both of those things can also be done in KNIME using their visualization tools. So, what you do with your pharmacovigilance tool, as I mentioned, it definitely has a great deal of flexibility depending on what your research question is. But with today's workflow showing how using KNIME, it can definitely be used as a pharmacovigilance tool to identify our special patient population. So I really do hope that you enjoyed watching this tutorial and find this information useful in your research questions.